AlphaFold is an AI-based method for predicting protein's 3D structure from its amino acid sequences. Compared with traditional approaches such as homology modeling and fold recognition, AlphaFold is faster and more accurate. If you're not familiar with this approach, you can learn more about them in one of our previous videos titled Protein Modeling. AlphaFold is available through an automated server that searches for templates and predict protein structures, requiring only a single click to initiate the process. First, I'll Google AlphaFold and go to its website. The only input we need is the amino acid sequence in FASTA format. Copy and paste the sequence in this field. Don't forget to select protein from this drop-down menu. If your protein has any post-translational modification or pTMs, you can specify them here. Then we click on Continue and Preview Job. What's the meaning of seed here? The seed is a starting number that controls the random aspect of AlphaFold prediction. Using different seed values can produce slightly different models, especially in the flexible or uncertain region of the protein. If you're not satisfied with the predicted model or if its confidence score are low, you can enable the seed option and generate additional models. AlphaFold will use different seed values automatically, which may produce a better prediction. Then we confirm and submit a job. Wait a few minutes while AlphaFold performs the structure prediction. Well, the model has been generated and we can see it here. We can rotate the model, move and zoom in or out by right-click, left-click, or scroll wheel of the mouse. If you want to copy or download the results, simply click on this button. This display option lets you customize how structure is shown. We can evaluate the quality of the predicted model using 4 confidence metrics: pLDDT, ipTM, pTM, or PAE plots. Let's switch to PowerPoint to explain these metrics, then we'll come back here and evaluate the quality of our model. pLDDT stands for Predicted Local Distance Difference. It's the most important confidence score because it tells us how confident AlphaFold is in the predicted position of each amino acid residue. The pLDDT score ranges from 0 to 100. A score about 90 indicates very high confidence in the predicted position of a residue, while a score below 50 suggests low confidence and may indicate an intrinsically disordered or highly flexible regions. In our model, we can see that most residues have a pLDT score above 90, so they're shown in blue. The color scale represents the confidence level for each residue. As you can see, most of our model falls within the high confidence range. PAE, or predicted align error, shows how confident AlphaFold is about the relative position of different parts of protein, rather than evaluating each amino acid individually. This is PAE plot. Dark green regions indicate low predicted error and high confidence in relative position of those regions. Light green, yellow, and red regions indicate higher predicted error and lower confidence. In our case, we don't see any yellow or red region, which suggests that the model has high confidence overall. The x-axis represents the score residue, and the y-axis represents the residue used as the alignment reference. For example, we may see 2 domains that both have pLDT score above 90, but the PAE plot shows low confidence between them. This means that the individual structures of the domain are predicted with high confidence, but their relative orientation is uncertain. pTM stands for Predicted TM Score. It measures AlphaFold's confidence in the overall structure of predicted protein. The score ranges from 0 to 1, and the closer it is to 1, the better. A score about 0.8 indicates high confidence in the overall fold, while scores between 0.6 and 0.8 suggest moderate confidence, and scores below 0.6 indicate that the overall structure should be interpreted caution. Our model has pTM score 0.98, that is indicate high confidence. And finally, ipTM stands for Interface Predicted TM Score. It's only used for multi-chain protein complex. It measures AlphaFold confidence in the predicted interface between different protein chains. A score about 0.8 indicate high confidence in the predicted interface. Since our model has only one chain, an ipTM score is not available. I hope you found this video helpful. If you did, please give it a like and subscribe to the channel so you don't miss our future videos.